And with that, I'm going to hand over to Nick for the next session.
Hi, welcome to the short talk session for the morning. I'm going to be your session chair. Uh, I will be standing over here for the presenters or sitting down, and I will stand up when you have about a minute left. We have roughly 10 minutes total for every speaker. So if you want to shoot for 8-ish minutes, so you get a few minutes for questions, we can hopefully keep everybody on time. And our first speaker is Justin Landis from UNC Chapel Hill, and he's going to be talking about utilizing tidyomics pipelines with, how do you pronounce this? plyxp to summarize expression informed by transcript similarity. So take it away.
Thank you. All right, I'll try and go ahead and get started. My name is Justin Landis. I'm from UNC Chapel Hill, and today I'll be talking about my package called plyxp and how we're using it for hierarchical clustering on gene isoforms. So to begin, I'm going to kind of just make a brief advertisement for plyxp. If you are a user of the summary experiment class or enthusiast of dplyr, then plyxp might be great for your pipelines. We support many of the core like verbs out of the gate, and it is just that, an interface for dplyr for the summarized experiment class, an alternative interface. I say alternative because we are not the first ones to create this kind of syntax or interface. That merit goes to the tidyomics group who made ID summarized experiment, and their package does more than just interface with dplyr. It supports other tidyverse packages. But their code is unique in that the code you'd use for a summary experiment is almost identical to how you use it for a data frame. The caveat is that sometimes you might not get a summary experiment out as a result. Sometimes in a summarization call, you'll get a tibble. And so you may be wondering, if tidy summary experiment exists, why did I create plyxp? And it turns out there's a few reasons. Where is it? There's a few reasons why I made it, but the main was efficiency's sake. Back in 2023, when I first started using tidyomics, I noticed there was a occasional unwinding of the summary experiment into a long form data frame. And you can imagine with the certain data sets you have and how big it is, it might just take a long time to do this. It's not necessarily a trivial task to do. But I should mention, as of fall of 2025, plyxp has been upstreamed into tidy summary experiment, so this is no longer the case. You should be avoiding this unwinding. If you do find it, please let us know. But to understand how plyxp avoids this unwinding, we first have to kind of understand how dplyr works. So dplyr does something called data masking, in which it's just another environment that's inserted in front of your current evaluation scope. And within it, they have the bindings for your data frame or summary experiment within it. plyxp takes this a step further. Instead of having a single data mask, we create. Is it not clicking? Okay, there you go. We create 3 separate masks, one for each part of the summarized experiment. So we have one for the assays, the row data, and the column data, and each one has their own environment context to evaluate within. With this separation of context, we need to provide the user additional syntax so they can refer to the correct environment they want to evaluate in. The default context is the assays, so you don't need any declarations there. However, if you want to evaluate in the row data or column data, you have to decorate your expressions with either the rows function or the calls function, which are just sentinel values. They are just syntactically there to help the user express what they want to do. The separation of context would not be very helpful without certain crosstalk between them. So we provide pronouns that allow you to pull data from other contexts into your current evaluation scope. And these come in 2 flavors. There's an as-is variant, because sometimes it's more efficient to just work on the matrix as is. And then we have the default variant, which is like a reshaped form. So sometimes there's no guarantee that your requested data is going to be in the same size as your current evaluation scope. And that's what the goal of the reshaped pronoun is. Unfortunately, I don't have too much time to just talk your ear off about plyxp and all the things you can do, but we did make a preprint, and I would encourage anyone whose interest is piqued to go and view it, or just come talk to me. I'll gladly talk to you about it. So I'd like to spend the rest of my time talking about transcript aggregation, or more or less aggregation tasks. In Bioconductor, there's plenty of disciplines that would use aggregation in like microbiome data sets. They would do it for feature aggregations in fluocytometry data sets, so we'll do it for sample aggregation. And the packages you might reach for to do this thing is tree summary experiment to represent these hierarchical trees on your data set, as well as using packages like treeclimr or the MIA packages to do the kind of analyses on your data sets. But since we created plyxp, we're curious to see if we can kind of express these summarization tasks with just the ergonomic and efficient code of plyxp. And to start, we kind of just wanted to reach for examples that already exist in the ecosystem. And we knew that at some point it was probably going to be I don't know where Anyways, it was probably just gonna be like a group by summarize task at the end of the day, but we didn't know exactly how to implement it. And by looking at some of the code from other packages, I realized there's probably two major blockers to get this done for plyxp. The first one is that we have limited access to the information encodings about the tree structure. This is not to say that plyxp is not compatible with tidy summarize experiment or tree summary experiment. It's actually compatible with any summary experiment derivative. We just explicitly don't have access to the row links or column links or like the tree data structure yet. We have to use the predefined context of the row data, column data, and assays, and hopefully in the future this will be a thing we can easily extend. But for right now, we just have to leave that on the back burner. The second blocker is no tidy way to evaluate or express your node level of summarization. I'll circle back to this in a moment, but first we'll kind of discuss how we sidestep the first one. And to sidestep it, we kind of just decided to make our own vector representation that we could just put into either the row data or column data. And this is just like a poor man's version of a tree summarize experiment, basically. It's a records vector, which is just a fancy way of saying it's two parallel vectors, one encoding the node of each element, and the other one encoding which tree that node is associated with. And also has attributes for all the trees that are on that vector. So it's a very compact representation, but essentially very inspired by the linked data frame from tree summary experiment. Just a representation is not always that helpful without some utility. So we have some utility functions to work with it. We have ways to get relative information like the parent or children of each node, as well as some meta information at the very bottom that allows us to transform any given vector into some summarization portions of it. So we can get all the inner node representations or all the leaves or just all the nodes in general. And so with that established, we're able to write some code to at least do the summarization tags in plyxp with little help. To begin with, we have a summarize experiment, and the first call we have is a group by rows. So we're declaring that in our rows we're going to group by gene, and then the first mutate call is to perform. I just realized how this kind of shifted, but we are going to ignore that's kind of offset. But anyways, we're going to extract the ranges out of our range summary experiment or the transcript ranges. And you can imagine these are 4 transcripts. Each of these blue bars are considered the exons. And this second function we're piping into is our own custom function called cluster tx ranges, in which we're just doing a pairwise comparison of the exon intersections to create a similarity matrix, which you can pipe into a distance function and then get a hclust object out. Our tree vector is kind of our constructor for our tree vector object, which just takes any like a phylum object or hierarchical cluster object, and then this maps our observations to the nodes that we can then jettison back into our row data for our summary experiment that we now have access to do our summarizations. Now that we have access to, or we declared all our actual vectors, we can actually move forward with doing the actual group by summarizing tasks. And here we're doing something that's kind of a bit cheating. Group by in its essence is just an annotation of your data. It's just a way to declare the partitions of which you should do each group evaluation at. And so we kind of wrapped the group by function in which this group by nodes expects a tree vector, and whatever you're explicitly declare as a result of that evaluation is the level of summarization you want to do. So here we're saying group by all nodes, and then now we create a new table of 7 groups where each partition is indicated. And so you kind of can imagine that the next summarize call will kind of follow in kind. It's just going to iterate through the groups, pull out every subset, and then create the do column sums on each subset. And at the end of this call, we get a longer summarize experiment of 7 observations instead of 4. And so I think that is all I have time to kind of discuss. Unfortunately, if you're interested in contributing or talking more about these tree structures, it's very much a work in progress. Hopefully, this is going to be how we extend tree summary experiment in the future. I also like to thank my lab and also like to thank Thomas, because otherwise I probably wouldn't be able to present today. So thank you. I'll take any questions.
You were perfect on time. So do we have any questions from the audience? Do we have any online? Looks like, okay. So just, there we go.
Hey, thanks for a really interesting talk. I love the package. I've read your source code twice. Fantastic stuff. You're also one of the few packages on Bioconductor that use S7 as well as Vector. So I've been brainstorming with with your code, I guess. How do you feel about extending Bioconductor, away from S4? Yeah, I do it as well. Okay.
But the thing about have a general extension of any kind of class with much more generic. I think it can be done. We just have. Okay, sure.
All right. We have a few seconds left, so I just have one question for you. As someone that came into R from not a programming language background, so can you give a brief, like, what do you think is the easiest entry point for people like that who want to get into dealing with like layering environments and passing things through different scopes? Like, what made that easy for you, and like, what do you think makes it easy for other people?
Fortunately, with R, there's really hard ways to crash your system. I've done it before, but at least when you're just creating stuff from the base stuff without getting into.
Great.
Encourage just practice.
Great. Thank you so much. All right. Our next speaker is, your name is in here somewhere. Artur. Yeah, right? Artur Sannikov.
Thank you. Yeah, perfect. So I will be talking about this R package called HoloFoodR that was developed by Thomas Bormann, and I wrote the case study and tested the package. So it has very, actually, where is the thingy? Okay, yeah. Doesn't work. Nice. Okay, it's very slow. So yeah, let's go to the agenda. So I'll talk what is HoloFood, what is HoloFoodR, and what problem it solves. Then we'll get to the results, a little bit of a case study, and finally to the discussion and conclusions. So the HoloFoodR is a Horizon 2020 European project. It basically was aimed to study the animal gut microbiota and how to improve the animal gut and the animal nutritional value for the consumption of by humans. And there were 2 animals studied, chickens and salmons. More than 2,000 individual specimens were studied. A lot of data was collected by molecular physiological measurements, genomics, transcriptomics, and other omics. And in particular, they focused on the hologenome, which is a set of genomes of the host and microbiota. So hence the name HoloFood. Yeah, and obviously a lot of other omics like metabolomics, transcriptomics, etc. So they created a HoloFood data portal, which has a lot of data, which you can access both through the web portal, but also download it with Python via JSON. And then you need to transform it into tables to actually analyze it, which creates a lot of problem. Because if you ever work with JSON data, you know how messy it is and how difficult it actually to clean it and to put it in a format that is nice to work with. So there should be a way to get this data and transform it into a common bioconductor formats. These two trisomerase experiment and multi-S experiment. Not going to explain what they are because you probably all know what they are. So this is where HoloFoodR comes in. And the basic workflow of this package is that we get this data and we basically transform it into trisomerase experiment and multi-S experiment, so we can have access to more than 2,000, not 2,000, like yeah, I think 2,000 packages of the bioconductor ecosystem. Then you can just start performing data analysis and data visualization straight away without jingling with these JSON files and tables. So this is what we did. The package is relatively small, has only 7 functions where you can search the HoloFood database. Then you can get the data. You can add the data from the metagenomic database Magnify and also from the metabolomic database Metabolites. So the results, the package is released on Bioconductor and the paper has been published. It was published in November last year. We did a little bit of case study. So we fetched the data using this package. We filtered, cleaned, and just did a little bit of data visualization of the data. We then tested associations between fatty acids time and treatment, between microbiome composition time and treatment. And finally, did some multi-omics. So about the data, the data was about the fermented seaweed additives that was added to salmon diet. It has a lot of nutritional value and pretty diverse microbiota. So the idea was, how can we improve it? Salmon for the human consumption. So the results were that we did not find any effect of the treatment on the fatty acid concentrations, unfortunately, but we did find some time effect. So the concentration of fatty acids increased at the end of the treatment. Then we saw also that alpha diversity increased at the end of the treatment. We confirmed it with beta diversity. As you can see, there is a difference between the start and the end of the treatment. And finally, we did some multi-omics data integration with multi-omic factor analysis, which is aimed to discover latent factors that underline the biological difference. So the idea was, is any fatty acid connected to any taxon? And I will focus on factor 2 because it captured the actual multi-omics, so fatty acids and metagenomics. And here it's very interesting because you can see that Vibrio and Alivibrio, they change in the same direction as EPA and DHA, which are good fatty acids for humans. But the interesting part that Vibrio and Alivibrio are actually connected to bad effects on the salmon meat, and they are relatively bad for the salmons. So it's interesting that they are bad for the salmons, but in reality then they increase the amount of EPA and DHA for humans. So discussion and conclusions. So holomic analysis relies on curated data. And the main purpose of this package wasn't the case study. It's actually to show that we can breach the gap between the data retrieval and data analysis. So the actual data retrieval was the smallest part of this workflow, and the largest part was actual data analysis. And usually data retrieval is like 70% of your time that you spend, and then you clean the data and then you try to put it in a nice format that you can actually visualize and analyze. HoloFoodR can also facilitate teaching because we often teach with some toy data sets, which are very, very simple. However, the problem is that they are nice to teach the beginners, but when you get into a little bit more advanced data analysis, then you need real world data that you can actually teach nice advanced statistical techniques. And then if you want to get metabolomic data, then you can use external tools like Notame that will be presented later today. I would love to thank the authors of this paper, the fundings, agencies, acknowledgements of my 2 groups and 2 of my friends who gave a very detailed feedback on these slides, and the HoloFood Consortium for actually providing the data portal and the data. These references you can read online. This slides are available online, and you can contact me and also read the paper by scanning these QR codes. Thank you.
All right, also great on time. So do we have any questions?
Hi, just a quick question about the results you were having, because you said Vibrio was coming up. You said it had a negative effect, right? Did you look any further? Because one of the more famous Vibrios is Vibrio cholerae, otherwise cholera. Did you investigate that any further and have any findings? That's good.
All right, do we have any other questions? Nothing online. We have a little bit more time. So I guess I'm going to ask another question about your taxa. So why did you choose to stick at the genus level? Did you not think, or did you not feel like you had the resolution to go further, or? Okay.
In my actual studies here,
Fair enough, fair enough. And then we have one question online about your logo.
I like it. I don't know where's the chicken. Maybe it was easy to find. Easier than salmon.
Very nice. All right, so if we could thank Artur and then get ready for the next talk. All right, our next speaker is Dattatray Mongad from Turku, and he's going to be talking about YAML-based workflow reproduction. So take it away.
Yeah.
Stand up when you have.
Yes. Thank you. So today I will talk a little bit about one idea which we have been discussing from a couple of months, making microbiome-based analysis simple, which will be based on some YML file. So the motivation behind that idea is I have been working in microbiome field, so I know the experience when the new data came into lab, and we all got excited, and then we designed one GitHub repo, start accumulating our code, properly documenting it. But when there are a couple of discussions and some the analysis gets on a little bit complex, and also the new data come in and there are many changes, and then analysis gets complex, and with it our GitHub repo and all our codes also get complex, right? And I have listed here some of the pain points which I have faced, like hidden parameter choices. For example, if we have multiple models in our analysis and if you want to change some parameters, then you have to go all through your code, see what changes should be made and make them manually. We like to save outputs in a scattered directory and name it by some dates, which doesn't make any sense. And unclear dependency is one of the pain point which I have faced, like on which R object my analysis is depend on. So I have to manually go through all the R codes. So then I have got again the inspiration from the SnakeMake, which I had working on SnakeMake workflows to pre-process all my raw data. So I thought if we can have the YML-based analysis plan, like the SnakeMake documents all the steps in the workflow, which workflow and the dependencies as well, then it would make the microbiome analysis simple. So the idea is like we will have a YML file. It will list all the analysis steps, input, outputs, and all the functions and parameters. And then we can design one package. That's for now we have named it MIA plan. And in this package, there would be some couple of R functions which will load this YML file and convert it into targets pipeline. It is kind of equivalent to SnakeMake in R, and then execute the whole analysis plan. So the plus point would be as you are saving all your analysis plan in a single file, and it is YML based, so it is more readable as compared to a long R scripts or Cortha documents. It would be easy to share, less scripting would be required, especially for the beginner, and reproducible, the important point. So what could the YML file contain? So uh, we are thinking of the most simple version of YML file, like it would contain the project name, input for each steps, and different functions and their parameters, and again the step order and dependency. So in most of our analysis, so output of one step will be the input for other step, and that way we can control the dependencies in our whole analysis plan. So we are thinking of the architecture like this. So there would be one user layer which we are also thinking of adding the Shiny interface, so that user can design their workflows with single clicks. And in this MIA plan core layer, we will design some functions which will load this YML file, validate it, and convert it into targets pipeline, and execute it in this layer. As I'm mostly working on microbiome analysis, this is the most simple analysis workflow which we use. So you can imagine that if we take an input and there are multiple steps, and like for example there are multiple kind of transformation and normalizations we can do, and for each downstream analysis we can use different transformations. So usually now tree summarize experiments can save multiple transformations, but we can have different variants of R objects in our long R scripts, right? And it is very hard to track the dependencies of each and every parameters and the effect of each and every change. So in this case, this kind of workflow can help. So expected benefits, the most important is reproducibility. If you save the plan into single file, uh, it would be easy to share in your collaboration, and it would be easy to reproduce all the exact outputs from the same analysis plan. Also for expert bioinformaticians, it would be easy to design some modules in your whole workflows. And for large consortium or cohort projects, the workflow would be too much standardized. And one of the main thing the targets can provide, the targets pipeline, is execution of multiple steps parallelly. So the HPC ready execution would be one of the benefit. So currently, as I said, we are in this phase defining the minimal YML schema, and creating this Shiny app for designing this YML file. And currently we plan to accommodate only few basic functions from the MIA environment and test this plan actually. And we are also thinking of adding the support for MIA dash. So uh, it is a web-based application developed by Julio. So in this application, you can just perform your analysis with simple clicks. So we can extend this execution to MIA dash as well. And one of the main thing is a plugin system. So if a user can have their own custom R codes, it can be accommodated into this workflow as well. And yeah, thank you. That are my acknowledgements. We 5 people, including me, we are just brainstorming on this idea currently. Thank you.
Thank you. It's nice to see lots of talks on reproducibility stuff. So I hope everybody else is enjoying it. Do we have any questions? Oh, we do.
What is advantage of this workflow managing compared to existing workflow management systems like Nextflow or SnakeMake? Or is it like more aimed to microbiome analysis?
We are trying to speak the RNA.
Do you know about Nix and Express, which might fit into this? Because it allows you to create completely isolated and completely reproducible environments, which is the next step, I guess, including system dependencies and R versions and everything. So you might want to consider that. It's not easy, I will tell you, but I might be able to help because I've been using it for a couple of years already.
It looks like that's it. So if we could thank Dattatray again. All right, our next talk is Juho Pelto from here. And I guess, is that you coming down? All right. Stand up. 1 minute. Yeah. Okay.
Yes, hi all. So I'm going to present a method for differential prevalence analysis, especially for microbiome studies. Okay. Yes. So first some background. So in microbiome studies, you can like at least in principle like measure the abundances of the microbes. But there are some problems with the abundance data. So first, it's compositional, and second, the distributions of the abundances, they are very they have like difficult distributions. There are many zeros, but then also some very large numbers. So it can be tricky to model statistically. So you can make things much simpler if you just transform to presence absence data, and so that just means that you just look at whether the abundance is 0 or just something above 0. And it seems that you lose a lot of information when you do this simplifying transformation, but actually some recent evidence suggests that you may not lose that much information, maybe more like just some noise. So that motivates us to conduct a differential prevalence analysis. And what that means is, in the simplest case, it's just like comparing prevalence of microbial taxa between 2 groups, like typical like between healthy people and people with some disease. And I use this simple two-group comparison here as an example, but this analysis can be like something much more complicated. You can have like lots of variables like covariates and also like interactions and things like that. And this analysis seems very simple, and it kind of is, but there are some things that can still be like optimized and may cause some problems. So first are these what I call here like boundary cases. So cases where the taxon it's absent in all samples in one group, or it's present in all samples in one group. And in that case, you get basically in infinite odds ratio in logistic regression, so that can cause sometimes problems. And the second thing is that, of course, there are like many hundreds of taxa usually in data. So you have to do some multiple testing correction, and that can be done in various ways. So to answer like these challenges, we develop this method DIPPER. That's still it's based on logistic regression, but like a central or like the core idea in it is that it uses it's a Bayesian method and uses a hierarchical prior for the differential prevalence parameters. So those betas there, and as the hierarchical models work, so the prior its width and here also the skewness is driven by data. And this leads to multiplicity adjusted or like shrunk estimates and intervals, and which are finite also in boundary cases. So this happens like automatically because of this hierarchical model structure. So you don't have to make any additional postdoc adjustment for multiplicity. So and because the prior is here the very central of this DIPPER, we can look at that a bit more. So as that prior, we have chosen asymmetric Laplace distribution, and this choice was mostly based on empirical observation. So this just seems to work quite well. And its mode it's fixed at 0, so or odds ratio of 1. So meaning kind of reflecting the assumption that most taxa they are not differentially prevalent between the groups. And we also allow we choose this asymmetric version of the Laplace distribution because we have observed that the differential prevalence effects they are very often like asymmetrically distributed. So to accommodate that, we allow also skewness for the prior. And those hyperparameters that determine the skewness and width of the prior, they have their own hyperpriors. So this is like what the model is, and then how it performs in practice. So here are like two illustrative examples. So first, there's a kind of null data. So this is the data where we just took a real data and created their random case control groups. So we get data where there are no like true population like differences. So all the ground root is always 0 for all of these taxa. So we can see how the methods like here we know the ground roots. So here we know how what the result should be. So if you run like a standard logistic regression in like in the in the central panel, so you can see that you get what kind of results you get. So you can the estimates vary around 0, and sometimes you even get those if you don't make any multiplicity adjustment. You also sometimes get those significant results. Even like the 0 is the true value here, but you get like significant results. So false positives sometimes if you don't do any multiplicity correction, and then if you do something like multiplicity correction, like in the rightmost panel, then you get rid of those false positives, but you also make the intervals come excessively wide, and you may sometimes lose power because of that. But what DIPPER does, it kind of automatically kind of recognizes that the differences in the data, they are just random variations, and the true effects are basically 0. So the interval, so the estimates are very close to 0, and the intervals are also very narrow. So it can very accurately estimate like 0 effects when 0 is the true value. Then what happens when what happens when we have like data? Here is data from study which studied colorectal cancer patients versus like healthy subjects, and what happens then? So here DIPPER can now it can like it can kind of recognize that now there are maybe some true effects, and it can find lots of potentially true differences. And then we can see now that if you just make some heavy multiplicity correction in in a standard frequentist method, you will lose many potential true findings. And those NAs there indicate boundary cases where you don't necessarily get any result if you just like run the standard logistic regression in R. So these are just two examples, but we also benchmarked this dipper on like very many data sets against some other methods. And here is just the error rate and like empirical error rate estimates based on data sets where we created random case control groups in data, so we can kind of simulate a realistic null data, and we can estimate empirically the error rate. So Dipper has this error rate like basically at the nominal level, but then we look at the just take some very many real data sets comparing healthy and like some people with some disease and see how many significant results different methods find. We compared this against some other differential prevalence methods, but also some differential abundance methods for comparison. And it seems that Dipper is very sensitive. It detects a lot of potential differences, and note that the scale is logarithmic there, so the difference is actually pretty clear, like two or three-fold difference between the least and most and least sensitive and Dipper. And then lastly, we also look at how the results replicate between studies, because on real data we don't know the ground truth, but this one way to kind of, if you can replicate the result between study, it's likely a true positive, and if you get conflicting results, then it's like false positives. And if you compare this, it seems that Dipper like outperforms the other methods in this sense. So yes, so this Dipper is a method for differential prevalence analysis, and it seems to be very sensitive, but still have like a error rate at the nominal level, and it also provides multiplicity adjusted estimates and intervals. So thank you.
I think we have time for one quick question, if we can get one in. It's got to be quick.
Very nice presentation. Can you say something about the what data you feed on, or have you tested it when? Because, for instance, microbiome, it depends a lot on the depth and the classification. What do you use? Very nice.
Great. If we could thank Juho again, and then get ready for our next speaker. All right, our next speaker is Ali Mostafa Anwar, and yeah, the clicker's there. I'll stand up when you.
Okay, perfect. Thank you. Hey everyone, my name is Ali, and I'm here as a second year PhD student here, based here in EuroBioC2026 in Illinois Coffee Group. And here today I'm presenting LimROTS, our differential expression bioconductor package for doing proteomics, and it's got published end of last year in Bioinformatics journal. So you can check more details. So in large-scale proteomics, we usually analyze thousands of proteins. We compare different biological conditions, and this data is usually high-dimensional and noisy, and also affected by biological variation, like covariates, or technical covariates, like different batches. And to take a look at a typical proteomics experiment, especially in a clinical setting, it usually have hundreds of patient samples, and these patients of samples have different ages, and age, if age is a risk factor, so you have to adjust for age, for example, during the differential expression analysis. But also we cannot analyze all of these samples at one shot in the lab, so we have to divide it into several batches, and this introduce unwanted variation. So you do this experiment twice with different set of patients, and you get different statistical significant proteins. And we believe that this is happening because standard statistical methods may not fully balance between three things: the statistical error, the reproducibility, and the adjustment for complex design. And this is why we created LimROTS, and LimROTS combining two statistical frameworks. It combines Lima, and we take like the strengths of Lima for like the flexible modeling, and also ROTS for the strengthening in optimizing the statistic reproducibility score. And in summary, like how LimROTS work, we fed the linear regression from Lima with the empirical bias moderation to stabilize the variance. We take all these results and then use the ROTS framework to reproduce this score. So we got like this new ranking statistics score that we can use it for calculating p-value and FDR. So the end result of LimROTS is actually p-value and FDR. So it's a statistical method. So to benchmark LimROTS against other methods, we used 20 standard spike in dataset. 12 of them were data-dependent acquisition mode, DDA, and 7 of them were data-independent acquisition mode, DIA. And we benchmark against ROTS, Lima, DQMS, MStats, DEP, SAM, and T-test, and ANOVA. And for the case study 1, this is like a simple experimental design. You have only 2 conditions. This is the result for the 7 data-independent acquisition mode. And here we applied a cutoff FDR of less than 0.05. And we didn't apply fold change cutoff here because we believe that, not believe, but spike in dataset is designed in a way that each condition have at least 2 fold change. So if you apply fold change cutoff, you artificially inflate your true positive. So we believe that it's wrong to apply a fold change cutoff here. So if we looked at the results for LimROTS, for the F1 score, the NMCC score, and also the BR AUC, we can find it very, very comparative to all the other methods, which is kind of establish LimROTS as a good differential expression tool for proteomics. But then to increase the complexity, we artificially induced batch effect. So we use the 2 sets of data sets. It is the same samples, but got quantified with the same software, but on different method, something we called wide DIA setting and narrow DIA setting. And these settings are producing a little bit of variation. So we use this as a batch effect. So we didn't do any batch correction here. And the batch effect term was modeled as a covariates when it's applicable. And now we can start see that LimROTS starting outperforming other methods in terms of the F1 score or the NMC or even the BR area under the curve. And as like independent validation is always like a valuable thing for the methods. A recent article was published last month by ELU lab group here in Turku Bioscience, and they discussed in this article how different pre-processing can affect the results for ROTS and LimROTS. And to take a look at one of the case studies there, we didn't find any statistical p-values reported between these differences in the method. We didn't find it, but we can still see that the performance of LimROTS is favorable over ROTS in many evaluation cases or in many evaluation matrices. But more results are there in the article. So to conclude, LimROTS combining Lima and reproducibility optimizing ranking, and it account for complex experimental design, improve the results from the omics differential expression analysis step, and also it supports summarized experiment and biopsy parallel for parallelization. And for sure, validation on additional data set is always valuable. So we are like welcome if someone want to test more if you have data. And I would thank my BIs, colleagues, and also the co-authors. And we have poster 31 here today for our new method for batch correction called NMF batch. So you are welcome to visit. Thank you. And sorry, I have to thank Whiskey, our cat, because she helped me in preparing the slide this morning. Thank you.
Thank you so much. So we have time for a few questions if we make them quick. I see. Oh, first hand.
Thank you very much. Very interesting. Maybe can you quickly give an intuition? You mentioned NMF batch and LimROTS. I think it's always nice if you can use your batches as covariates in the estimation.
Yeah. So I think we believe it's for sure you have to correct for your batch effect, but we believe also that the batches as the term as in your covariates without doing any batch correction, because most of the batch correction, at least for proteomics, actually removing variation and not differentiate is it a technical variation. But if you treated the batches as like a covariates, the model will account for this directly without the need of remove. But we animate batch in a way because after doing differential expression analysis,
Great talk. Does the user need to perform batch correction before using LimROTS?
after differential expression analysis, then you have to do batch correction. Don't use this after that you can tweak your
All right, I think that's what we have time for. So if we could thank our speaker again. All right, our last speaker for the session is Rishi. Yeah, thank you. Is Rishi Das Roy. He's going to be talking about benchmarking neighborhood-aware DEC. So.
Yes, thank you. Okay, so let's see if slide is working. Yes. So now I'm you and between the coffee session, right? So that is a great opportunity that you can ask me more questions. No one is behind me. And I'm not only probably in between coffee and you, but probably in between your favorite tool for differential expression genes, or am I? Let's see. So I work in a biodata analytics unit, which is a core unit of University of Helsinki. And I have to provide data solutions to many groups and their different settings. But before that, I was a postdoctoral researcher in a group where we were studying like tooth developmental biology. And there I published this article, DELocal, for differential expression gene. Why I did that? Because when we were trying with other tools, my colleague, she was not very happy with the result. And we've shown that the information we get from DELocal, that is very different from the other tools. It is not like it is better, it is different. So how it works and why? What is the motivation? So Auti, my colleague, what she has done, like she gone through many literature and curated the list of genes which affect the development of tooth. And she was very sure that these genes differentially express between these developmental stages. And here, like a few of these genes example, you can see like they are differentially expressed like visually, but with the nearby genes, they are not differentially expressed in between these two stages, birth stage and cap stages. So this motivated us, like she was saying, oh, these are like differentially expressed to me at least. So can you catch it? Okay, so I said like, okay, let's just develop a method where we will compare the gene's expression, which is neighboring gene, and let's see what happens. And then we found this one. And so I published it, but I was not happy with myself that will it work for other organs and other conditions? It may be just a good luck I have. So yeah, so then the objective was to slow. Okay. Yeah. So the objective was to benchmark DELocal in a diverse data set. So the challenges was there is absence of comparative tool where I can easily compare different methods. And I have to do it like repetitively because in my future projects, there will be coming like more bulk transcriptomics data. And I have to do this comparison. And also like my collaborator will always ask me, why not this method and why not that method? So I wanted to make a better system where I can do, if not DELocal, some other analysis too. And then there was an absence of gold standard data, like how to evaluate these methods. That is always scare. So first, the challenge is like this comparative tool. So in the left side, we can see like during this last decade, there have been like many tools and they have developed many input format and output format. Like now, I was looking at the summarized experiment, how it has so many different summarized experiment now. Anyways, like so in my tool, you have like this summarized experiment as an input, and then you can do many transformation, VST, log, CPM, R-log, and then you can have like ggpubr, which is produce publication quality figures. And so that is really helpful for me for my future projects that I can quickly do it. And then there are like differential expression methods which you can choose here and can compare, and the result in the data frame format. Like if you see in the output, it is very hard to compare all this, and that's like quite not motivating us to use any other tool. If you have a standardized pipeline, you got it, just keep doing it, right? So now, like first example, like you can easily explore with broadSeq many different clustering algorithms like hierarchical clustering and heat map, and annotate it very quickly, whatever you information have stored in the summarized experiment. And in the right side, you can see like this, you can use MDS plot with VST. MDS is from Lima and VST is from DSEC2, and then PCA plot with log CPM. You can play with the data, and also you can do advanced PCA exploration. Oops. Sorry. It is not going back. Okay. So we can do advanced PCA exploration also with this. So now when I develop this tool, come on. No, it jumps. Okay. What is happening? It doesn't have battery. Stay there. Okay. So when I did this with functional programming, it's like everything is oriented to functions, and which helps because then I can add a new function for differential expression with this functional programming, which is just a wrapper function, and I can pass this function as a list. And when I compared, so you can choose which functions to compare and which functions to remove. So in future, if you are going to use it, you don't have to compare always. So you can just use 2 or 3. Right, and that is like another approach I use. I think that is quite nice. And now go back to the benchmarking step. Move. Okay. Stay. So here it is like the data set I have used. So in this data set, what we have from mouse, like brain, heart, liver, kidney, and from different developmental stages. So there were like 2 primary contrasts I used for each organ. So first I tried to find like which genes are upregulated compared to the rest of the organs, and then which genes are upregulated in the mid-developmental time point compared to the earlier time points. There are 2 different. And then if in an example, if in see in heart, so in case of heart, then after identifying the upregulated genes, I took top 10 genes from 10, 20, 50, 100, 200, 500, and I was trying to look for new information, which means like if there is a new gene there, which I've identified, so try to find this with the Jaccard index to find the similarity if these methods are similar or not, which is like you can see in the bottom, like DELocal is like least similar. If we look at through these Venn diagrams, again, like if this is like top 10 genes or top 100 genes from each method or top 1,000 genes, you can see DELocal is standing out. And for if each contrast for each organ, we see like these methods are the rest of the method, and DELocal is identifying always like in out of 175 genes are unique genes, new information. Are they real? They are any makes any sense because they are not going with any other methods. What are they? So I don't have any gold standard data, so I use gene ontology. So here you can see in the left side, there's an organ level when heart is compared to the rest. So it gives same gene ontology as other methods. So the information is same, but new genes. But next in the developmental stage, you can see like it has identified cartilage development and connective tissue development, which are like totally related to heart development. And it has been not identified any other method. So next is like I tried, okay, that is gene ontology. And what about if I try something new? So I use kidney cancer data set and with disease ontology. And disease ontology has never been used in benchmarking anywhere. And here you can also see like it has been uniquely identified nephroblastoma, which is not identified anywhere else. And then like in the right side, you can see like I was thinking like why this, what are these genes, what they do. So I found like they are like highly expressed genes and little bit less log-fold change, because when the expression level is high, log-fold change cannot be high. And so in summary, yes. So with what happened with the rest of the organs I tried, so the equal sign shows like you get same gene ontology terms, and where it's cross, you get bad gene ontology terms which are not relevant to that organ, and smileys makes these are very relevant for the biologically for those organs. And so this has been done with the human and mouse data, not simulated data, gene and also disease ontology data. So DELocal provide you different unique genes. So if you are looking for something different, so try DELocal. And then biologically relevant terms. And no AI used while developing this because it was not there till. Probably if I develop now a package, I will do that. Okay, so that's it. Thanks.
All right. As someone who's worked in a place without gold standards, I can empathize. So it's very nice to see this. Do we have any? Oh, okay. First question.
Hi, good presentation and interesting approach. So I will start off with I'm slightly skeptical, so I want to talk through this so you can convince me. The thing when you're comparing DE-seq2 and all these other methods, they all use different underlying models, right? And there are DE-seq2, Lima, they all use different underlying models. So that's why they often get different results, but you get quite different. So what underlying model are you using in your package? So to reiterate the question, what is, so I know, I understand now that you're doing it based on the two genes that are related to each other, but what's still the underlying model are you using to get your effect size and p-values and stuff like that? Okay. I can ask again at the coffee. It was just more of like, do you use a linear model, negative binomial? Oh.
All right. I think it is time for coffee, but first I think I've been told Tuomas has an announcement for us. But before that, can we?
Hi. How can I? Great. All right, so everyone, my work is on mapping the host microbiota interactions across the territory of Finland. And to give first a context, a historical context for why we are performing this work. One of the first geospatial analysis of epidemiology done in the world was from John Snow in London. He mapped out the cases of cholera during an epidemic in Broad Street in London, and doing that, he was able to verify that there was a pump in that street that was responsible for the epidemic. By discovering this pump, he was able to provide evidence to the authorities, and they removed it and solved the sudden epidemic at the time. So this is basically the motivation behind our project. And why are we going to apply this to the gut microbiome in humans in Finland? Well, we already know that the geographical location of an individual is usually the most important aspect when it comes to the variation in their gut microbiome. As we can see in this graph on the right, this shows us an ANOVA regression where province was the most important variable for the variation across the individuals. So our objective is to model the distribution of the gut microbiome across the territory of Finland to see if we can find any patterns that can be uncovered and deduce certain causes or at least a correlation between diseases or any kind of environmental factor that could allow us to understand better how the gut microbiome is influenced. So our method is basically to apply the FinRisk data set. It comes from a health survey from Finland that was very recently had stool samples, had the metagenomic sequencing done to them. And now we have basically around 7,000 samples from individuals almost across all of Finland. But there are some gaps, especially in Middle Finland. And we need to overcome these gaps by applying some statistical tests. And we are going to do it using basically a Bayesian approach, using the INLA R package, because it is very efficient, very computationally efficient. And we are going to apply it to a grid that will allow us to model the distribution of the gut microbiome using a linear regression, basically. So how do we do this? First, we have to agglomerate our data into genera, because using all of the highest taxonomic level is a bit cumbersome. It leads to a lot of zero inflation. It leads to a lot of missing entries. So agglomeration usually reduces the amount of variables that we need to analyze and also reduces the amount of samples with zeros in them. Then we perform a normalization, a relative abundance normalization, to account for sequencing depth. After that, we perform a selection for the genera that we find that are geographically significant. And then we perform a normalization to get a Gaussian distribution that is then modeled using INLA. Can anyone advance? Okay. So how are we going to do this? We are going to make two different distinct Bayesian models. One, a very simple one, accounting for age and sex, because we already know that these covariates are very important in the variation of the gut microbiome. And another one that adds this spatial graph. Doing this, there's going to be a variation in a parameter called deviance information criterion that determines if the added complexity of the model is significant and valuable for us. So it will cause a reduction in this value, and by this reduction, we can measure whether our model is good for our data or not. All right, so here we are doing a selection of the geographically significant taxa. What we did here was apply a Gary C geographical test to evaluate whether they have a good spatial distribution that can be evaluated using INLA. And the blue dots are the ones that are statistically significant, and we selected the ones that had at least a 10% prevalence in the samples. And the 14 highest ones, because there was an arbitrary choice to get just a good enough amount of samples to be analyzed, of genera to be analyzed. And here are some, thank you. Here are some of the results. These are the maps for the prevalences that we obtained across Finland. What we can see is the red regions are the ones that have a higher than average spatial distribution, and the blue ones are the ones that have a lower than average gut microbiome distribution. So these are all different genera, and what we can see is that a lot of them have a higher than average presence in Lapland, around Kuopio and Helsinki. That's something that we find very common among the genera. Can you go to the next slide? And what we can conclude is that the geographically meaningful taxa, they appear to have a very strong correlation between each other. And these metropolis are the ones that have a good correlation between each other, except Lapland, that is the largest region when it comes to this higher prevalence in genera. And our next steps is to check the sensitivity of our method by evaluating each one of these regions independently, and also to check the distribution of the taxa to see if they correlate with chronic diseases. So I'd like to thank my cohort, my doctoral network, Hologen, and also my supervisor, Aki Havulina, who has helped me throughout this project, and the doctoral Department of Technology in general. Thank you.
Great, so there's a lot of time for questions. There was one.
Thank you for the presentation. Going like 2 or 3 slides when you show this map of Finland with a very sharp divergence between there's populations in the north and then in the south, with the exception of Helsinki. Would you say that this is due to difference in diets within the same country? And second, do you measure any difference into clinical implications on all this? Thank you.
We are doing some analysis now to understand why this is happening. What we discovered, this is an analysis that we are still analyzing, is that our data set contains people older than 65 in those regions specifically. And what we saw is that they tend to have a higher alpha diversity when it comes to their gut microbiome. We still are not sure why exactly that is. It could be because older people usually take, for example, more antibiotics or proton pump medication that are associated with certain dysregularities in the gut microbiome that might increase the alpha diversity. So this could be the explanation behind this higher variation for certain genera in these regions. But we need to investigate this further to check how can we approach these results.
Great. So in the interest of time, I guess we'll thank the speaker again and move on to the next speaker. I'll try to keep you right on time for lunch, I promise. So we now have Ville Laitinen. So hopefully the clicker is doing okay. It may be proximity to this computer. Okay, so that works. Thank you.
So good morning. I'm Ville. Prospective microbiome modeling. So the motivation for our work is in the fact that most microbiome studies so far have been either cross-sectional or longitudinal analysis, but the prospective dimension is still largely unexplored. However, like suitable data sets are starting to accumulate, some of which I've listed here, and in the like based on this data, several reports of like associations between the microbiome now and the health event in the future. Ones that I've listed here, like the titles of the articles are, they're all based on the FINRISK data, which is a Finnish cohort. So there are some studies, but the methodological best practices are still like largely there's no consensus on it. That like for that reason we're trying to do a effective assessment of the field, like what sort of methodological approaches have been used and so on. Very quickly, the prospective setting means, at least approximately, we have a time until some event which we try to associate with the microbiome and possibly some other code. I'm trying to illustrate that setting here in the figure. So in the beginning, we have a population, study population. We take a microbiome sample, then we follow them in time until the end of our follow-up period, and then we observe them, monitor the subjects for an event. Heart failure here. Subject 1 is in heart failure at time 5 is heart failure at 5. The rest of the subjects do not, and this means that their data points are censored. This censoring is, of course, a characteristic in prospective and level modeling. So that's the basic idea. And here, like we've done a literature review on which methods have been used in active microbiome studies so far. I have collected the methods here in the table. Unsurprisingly, Cox regression is the most widely used method. Interestingly, these methods that do not account for censoring, the first row, have also been used quite successfully to provide a robust association signal. Also, interestingly, what is absent in the table are many of these more modern machine learning methods like deep learning. So definitely room for expansion of this. But yeah, so the rows tell you whether or not the method accounts for censoring, and the methodological considerations. So when you're doing prospective microbiome analysis, you of course have the typical microbiome-related challenges and issues like high dimensionality and data characteristics like compositionality and choosing which feature representation to use and so on. Then you have a different set of challenges in survival analysis, prospective. It is to model selection and validation. But then when you put these two together, there are some other challenges or open questions that are emerged. For example, the fact that the microbiome is highly dynamic can cause, questions. Dynamics themselves may contain important prognostic information, or dynamics may mask associations. That's something that I've tried to illustrate here in the figure. Top panel, we have health in some units. At the end of the follow-up, abscess and results in an event. And below, in the lower panel, we have the microbiome state in whatever. The brown diamond signifies the baseline sample, and that's the only data we have of the microbiome. The microbiome trajectory that we have in gray. Now let's assume that we know for a fact that on the population level, the lower state would be associated with the event.
example of the upper state, this particular data instance actually asks the true association. Other interesting aspect is like defining the relevant time horizon. So presumably like different microbiome representations picked different events. Times. Of course, this type of like association decay is possible, but it's also possible that there are like sweet spot windows for predictive potential. So, based on the FinRisk data, I have one teaser demonstration here. So what I've done is fit box regression, increasing time horizons, using all-cause mortality as the endpoint, like event, and genus level abundances as the predictors. And here I selected 3 sort of qualitatively differing first genus. We see that the model box regression provides statistically detectable signal at around 8 years, starting at around 8 years. Second genus, there seems to be a window for like optimal prognostic potential. Final, the third genus, there's a effective effect lasting for things going on. This is just a teaser. So in terms of bioconductor, like the survival analysis implementations, there are some packages that provide. Here I collected some, which are, but as we can see, it's most like box regression and Kaplan-Meier analysis and some model comparison tools, but quite a limited selection of methods and nothing directly directed to microbiome analysis in particular, but we're working on integrating some of the more common and not so common survival analysis tools in the media package, which is thanks.
Great. Yeah, we'll have time for one question.
Hi, it was a nice presentation. You nicely showed how the problems you might have come across, but I found it interesting that you talked about that the effect could be non-linear, right? Because you have this up and down. Well, then you want to use a Cox model, which then is assuming the proportional hazard remains the same throughout all the follow-up. Why not try to do something like a flexible parametric model where you can actually account for variation throughout the time?
It's like looking at what people have been doing so far, and just trying to illustrate that this supersedes.
Great. Let's thank the speaker again. Here you go. And this one, you point at that computer. Thank you. So our next speaker is Sneha Das, coming from Turku, talking about multi-omics survival modeling for incident disease risk prediction for prospective cohort studies.
Hi everyone. Today I'm going to talk about multi-omic survival modeling for incident disease risk prediction in cohort studies. Motivation behind our study. So as we all know that complex diseases are driven by multiple biological factors which are interacting with each other. Actually, multiple factors, even environmental factors. But in my study, we limited to biological factors. And integrating such heterogeneous data sources can help us better predict the disease risk rather than just looking at a single factor. And also availability of well-phenotyped cohort data with survival analysis data also helps us in improvement of the disease risk, and survival analysis data is just the data till the occurrence of an event of interest occurs. Event can be anything such as onset of a disease or just any event of interest, and the survival data is calculated from the baseline where the participants where the study began and information was taken from the participants to the end of the follow-up period. But there are some current limitations to the study. Some research gap is that most of the risk models rely on single omics layers that can limit your predictive capability. And then there are some multi-omics based models, but they often rely on a simple concatenation of the heterogeneous data sources. The problem is that that might not account for the inherent characteristics of the different sort of data sources. So to bridge this gap, we work with Integrated Learner, which is an already published tool from Prof. Himal Malik's lab. It's a multi-omics prediction and classification tool. And in the current work, we extend it to enable enable the support for time to event or survival analysis data. And Nalin from Prof. Himal's lab is actively working on developing the project, and I am testing the survival analysis model. So this is the workflow for Integrated Learner. It begins with taking the different heterogeneous data sources as inputs. After that, we have three types of data fusion techniques. Early fusion. So in early fusion, the machine learning models in early fusion, all of the different data modalities are simply concatenated together into one big matrix, and the machine learning model runs on the matrix as a whole, but does not account that those are different data sources. And then it gives the final prediction. Then we also have late fusion, where each of the modalities are trained separately using separate models, and then the final predictions are combined. The model also assigns weights to each of the omics layers, and the predictions are multiplied by the weights to give the final prediction. And then we have intermediate fusion, which is somewhere in between early fusion and late fusion. So in intermediate fusion, first, all of the modalities are trained separately to give survival grid from each of the modalities. And then those grids are concatenated, and the final model is trained on the concatenated grid together. So the modalities are also treated separately, and also there is a joint learning phase. And Integrated Learners has support for 4 types of outcome. For example, binary outcome, continuous outcome, multi-class meaning more than 2 outcomes, and survival outcome. In the last few months, I have been testing the survival model. So my presentation is going to be more focused on the survival outcome. And after running the model for survival outcome, we have 3 performance metrics to evaluate how well the model has performed. Concordance index, which is basically the ranking across all time points. Say, for example, across a follow-up time of 20 years, patient A got the disease before patient B. So C index is going to tell you, did the model correctly do the ranking? Integrated Brier score shows you it is a calibration of how well the predicted probabilities are in relation to what actually happened. And AUC, or area under the curve, is a representation of the discrimination at a specific point in time. For example, say at year 5, patient A got the disease, but patient B did not. So did my model correctly predict that? So data, so we have tested the survival model on the FinRisk data, which is a cohort of Finnish population sampled all across Finland. It has nearly 7,000 participants, and follow-up time of 20 years, has a very balanced sex ratio of 55% women and 45% men. And it has tool sampling data. Along with that, it also has questionnaires and other clinical outcomes, and the survival outcome as well. So this is the workflow for training the Integrated Learner on the FinRisk data. So we have tested it on 2 endpoints, the liver disease and all-cause mortality. And we for multi-omics profiles, we have taken metagenomics data from which we take taxonomic profile, functional pathway prediction profile, metabolomics profile, and antibiotic resistance profile. Then we do a pre-processing where we apply prevalence filter of 10% and detection threshold at 0.1%, followed by data transformation using log10 and pseudo count because the data, all the data sets were skewed. So to apply some sort of symmetry in the data, we had to do the transformation. And we did normalization using Z-score to standardize the features because the metabolomics has the features in gram per liter and millimole per liter, but the metagenomics profile had the features in relative abundance, so to account for that. And then finally, the integration and training process occurs using the 3 different fusion techniques. For survival modeling, we have several models available, such as xgboost.aft, xgboost.cox, GLMnet, random forest. For the current work, my results are based on xgboost.aft because we had tested it once on all-cause mortality, and we saw that xgboost.aft is the highest performing among all the other models. And also, the data is before inputting the data into the model, it is split into 70% training data and 30% validation data. And then inside the model, internal 5-fold cross-validation occurs where the 70% training data is again split into 5 different folds, and the data trains on 4 folds, and it validates on the left out fold. And then, yeah, as I already mentioned before, we have performance matrices with C-index, Brier score, AUC, and it also gives you the top featured hits from all the features that were taken. And this is a result from the performance of the model on liver disease and all-cause mortality. As you can see in liver disease, all 3 of the fusion models, the intermediate, late fusion, and early fusion, have outperformed the performance of the single layers. But yes, among the single layers, metabolomics is still shows nearly equally good performance as the fusion models. In all-cause mortality, we see that intermediate fusion does not perform that well, but early and late fusion are performing better than the single layers. But here also we see the same pattern with the metabolomics data. And yeah, for the next steps, for right now, I have only been doing a very random 70-30 split, but in the data set, we have a 55% and 45% women to men gender ratio, and we also have other covariates like age, BMI. So to account for all of that in the training and validation set, I plan to also do a stratified split that will ensure that the ratios are same in both the training and the validation set. We are also going to test it on a subset of features to see how performance will vary with feature counts. We are going to test it on different follow-up time intervals because these results are from 20-year follow-up time, and I think that's a very long time, and some of the biomarkers may lose their signal over such a long time. So we are going to try different windows like 10 years, 15 years, and so on. And then we also have to test it on other endpoints like type 2 diabetes and sepsis. Then we plan to do ablation analysis, which is just trying a combination of the different omics layers, because as we saw in the previous result, although metabolomics is performing equally good as the fusion models, but the fusion models are much better than the other 3 single layers. So we just want to see if we exclude metabolomics and try combinations of the other layers, then how is the model performing? And yes, the tool is currently available as a package in GitHub, but we are going to publish it as a bioconductor package, and it is going to have support for 3C object and multi-assay experiment slots, which are the standardized data science slots. And yeah, that's it. I have to thank my PI Leo, my teammates Thomas and Mulu for their support in this project, to Prof. Himel and Nalin from his team for his active development in this project. Yeah.
So we're a little tight on time, so maybe in the interest of time, you could find Sneha during the lunch break to ask any questions. So we'll go straight. Thank you. This one, this clicker. So our last talk before lunch is Stevie Peterson, who's going to be talking about transmogar, modifying the reference transcriptome for analysis of Australian indigenous population.
All right.
It's that.
Yeah, thanks. I'm just trying to do a test run so I can get this right. So thank you for all having me here today. I'm Stevie, and I'm working on a project back in Adelaide called the Prophecy Project. And we're looking at type 2 diabetes and complications in Aboriginal Australians. And really the motivating factor behind what we're doing is that the mortality rate is 10 times higher associated with type 2 diabetes in the Australian Aboriginal population compared to the non-Aboriginal population, and the complication rates are through the roof in terms of cardiovascular disease, chronic kidney disease, and diabetic retinopathy as just some examples. So the Prophecy Project is a completely Indigenous-led project. The questions come from community, not from scientists, which is fantastic. It's run out of Adelaide and with some of our team in Perth. Adelaide is historically the lands of the Kaurna people, and we're all very grateful to be working with the Kaurna people and on their lands. And so with the project, we have nearly 1,400 participants from South Australian Aboriginal communities. You can see Adelaide on the map. I think we've seen enough maps of Finland today, so here's a different one. South Australia is the bit down the bottom in the middle, and all of the participants come from the South Australian communities. In terms of our multi-omics, we've got DNA, DNA methylation, RNA, proteomics, lipidomics, metabolomics, inflammatory panel, ECG scans, retinal scans, a whole lot of clinical and psychosocial measures. It's a huge data set, and I'm primarily focused on the transcriptomics layer, the RNA. In terms of genomic variation with the Indigenous Australian population, 25% of the variation is completely unique. It's not seen in any other populations throughout the world. And a question that I had to ask as we were developing our pipelines and planning out the project was, if these variants or if these genomic changes are, or unique genomic features are driving the progression of disease, will we see it in the RNA-seq? Is it going to make a difference? I don't know the answer to that, so we had to figure out if it would. I don't know if anyone's familiar with star consensus, where you can provide a haploid set of consensus variants if using star and any other post-hoc counting methods, but nothing existed as far as I knew for using salmon or Callisto. So the question I wanted to ask was, can we bring the reference transcriptome closer to those of that of our participants? And that was very well received by community. They were quite pleased with that as an idea. And the question is, will it have an impact on the results? We can see that expect that it might have some technical improvements, but will it change the results at either the gene or the transcript level? So I put together an R package to do this, and thanks to Bill Watterson, of course. Thank you. Yes, the main function is transmogrify. And so we can take a set of variants, doesn't matter if they're SNPs or indels, and in a VCF, I built in some checking steps because no matter how strict your pipeline is for variant calling, it's going to be a mess. And so there's some checks there to make sure that when you're going to modify things, you're not putting in a SNP and then removing it with an indel or something. Those kind of things happen. So I can modify the genome sequence with these indels, which means the coordinates change. And so the package will track the coordinate shifts after modification, so you can put in the variants and then know where things have shifted to. And you can also add tags to sequence IDs so that you know what has been modified. And you can also modify the transcript sequences. This is the key step that I really wanted to do. And you can tag those to whether with what type of variation has gone into that transcript. I've also included a function, Mike and those working on TXR called Digest Salmon, where you can parse counts and get a nice summarized experiment out with TPM effective length and over dispersions. And I particularly built that for when we have divergent references. Say we wanted to give our male participants or our female participants different references, then this is specially written to handle that situation, or as we'll see in a minute, personalized references. Okay, I'm clicking the right button every time. I'm so impressed with myself. So we, as we were developing our methods, we had some six samples which we used some alternative library prep strategies on, and so we used that as a pilot study because we had the variants for those participants, and so I built personalized transcriptomes. I was still haploid, so I chose all of the sites where there was homogeneous for variants, and then I just randomly chose 50% of the heterogeneous sites. And I considered this to be the best case like reference you could get for these individuals. It's still haploid, so it's not perfect. And then I said, just using the salmon or I was using salmon, so using the salmon transcriptome for these personalized references, comparing it to GRCH38, the AFR subset from 1,000 genomes, across all of the subpopulations in 1,000 genomes, and then our prophecy consensus transcriptome, which used the same strategy of any variant seen in more than 50% of our participants who are not related. So here are the variants that I stuck in, and as you can see, when we compare the 1,000 genomes data sets, the pan-human and the African and prophecy, there's a huge amount of variation there, which is in common, some which is quite unique to the African population, and some which is quite unique to our prophecy cohort. And so I created these consensus references and our personalized references, and we're able to compare these consensus references to the personalized ones. And so when you look at the technical metrics about what happens in salmon, you can see that I want to face you and the screen. That you can see there's a decrease in assigned fragments as you move further away from the personalized transcriptomes. And you can see an increase in alignments to the decoys, and then an increase in alignments that were below the threshold. So we can take this as telling us that technically aligning to a personalized reference is good, and then these are the references which move further and further away, very as expected, and very much in keeping with what was shown in the STAR consensus paper. And then if we look at the similarity of the counts that we get, we can see that they're really quite similar in the same order as we would expect. You know, a bit of variation between individuals. I did all the non-zero ones that were consistently non-zero and the ones I'd call detected, just using counts. And you can see that the prophecy consensus looks the closest to our personalized references. So then I went and had a look at the overdispersions, and there was no global shift. I don't know if that was a surprise to anyone, or I had to think about this long and hard, and I was pretty comfortable with this. But then we look at them. Now I'm actually switching the viewpoint. The overdispersions on the x-axis are from GRCH38, and I'm now stepping backwards away from GRCH38. And if we look at either the prophecy or the personalized ones on the bottom row, that there's a little bit of wiggle, you know, around the zero line, but there's some genes, this is transcript level, I've just labeled them as genes, that change quite profoundly with their overdispersions, and that's probably the key to notice. So then what I did is I performed differential gene expression analysis, just type 2 diabetes versus not type 2 diabetes. We had a pre-sequenced cohort of 93 from some collaborators had done a specifically tailored experiment they wanted to do with some of our participants. And so I just stuck them in the standard EDGAR GLM fit methods, and I performed differential transcript analysis using the methods developed by Pedro Bardoni and Gordon Smythe, where you scale the counts by the overdispersions. And like I said, I did this a couple of years ago, so some of the newer methods that have come out I haven't done yet. Oh, you're standing up, Michael, thank you. Do the cohort level results change in terms of actual differential expression? At the gene level, no. However, at the transcript level, things get really different. As you can see, we've got p-values and log fold change on the diagonal there. Most transcripts don't change, which is what you'd expect, you know. But where there is significant changes in the variant structure on the genomes, you can see some things appear right next to the y-axis for the prophecy consensus, which were not seen earlier, and some things disappear. Some of that is driven by these overdispersions when we're dividing the counts by the overdispersions. Some of it is due to different log fold change estimates, as you can see in the panel on the right. So I think that's enough from me, and I'd like to thank everyone that worked with me on this.
We'd love to ask you a bunch of questions. So maybe you can let's meet us at at lunch for more questions on this really interesting analysis. And I'll hand it to Thomas for any late breaking announcements.
Yes, so this is the split of the rooms for workshops. So in Presidentti there will be Tomi, and in Ministeri Lieven and Tellervo there will be Johannes. Clear? And then the next slide is lunchtime. So hyvää ruokahalua everyone.
Test. Hello. Yeah, it works.
This one, I think if it's red, doesn't mean it's working. Yeah.
Try. Hello. Test. I don't.
That's the word. Yeah.
Yeah.
Actually.
What?
The pointer.
The palm? It works.
It works.
Yeah. The pink one that we missed.
Yeah, less flashy, but still like yes. Of course, yeah. And then I think tomorrow is going to be a black one, but not going to be everything.
Yes, let's see what. Cheers.
Hello, everyone. So welcome to this the last session of short talks. So we'll have 3 talks. Each is 10 minutes. And I will stand at 8. So let's welcome our first speaker, Fabrício Almeida Silva, who will talk about graph experiment.
Thank you. This works, right? Yeah. Okay, so this is one more package to the family of experiment packages. So you all know, I guess, summarize experiments and the packages that extend it. So yeah, summarize experiment is a core data structure which has assays, call data, and row data. Yeah, over the past few years, we've seen many packages that extend summarize experiment somehow. So for example, we have single step experiment that adds dimensionality reductions, so the reduced dimension slots, spatial experiments, for example, with spatial coordinates and images, tree summarized experiments with trees, so phyla objects, and multi-assay experiments for multi-omics data. But then I quickly checked on Bioconductor on reverse imports and reverse suggests, and this is a bunch of packages that, so IMC, DD, so from National, by the way, Loom, variant, QTL, reduce, MS, ragged experiment. So it's a family of packages, and the family is growing, and it will grow more, I guess. Networks are very popular in biology. So Susan Holmes once said that biologists are obsessed with networks, and I'm one of them. I'm a biologist obsessed with networks at the feature level. So if you think of assays at the row level, you have, for example, if you think of transcriptomics, you can represent co-expression networks, regulatory networks, co-occurrence networks for microbiome data, co-abundance if you're working with mass spec data, so abundances. And at the observation level, you can also use, for example, if you're working with single cell data, you can have cell-cell communication networks, cell-cell distances that you can then use for clustering and to identify cell types. If you have, for example, species in columns of your assays, you can have networks representing how species relate to each other. So it's really, really flexible. You can do a lot of it. For those of you who know single step experiments well, you probably know that there's this lot that is not so popular. It's called row pairs and call pairs, and there you can store pairwise relationships between features or observations. The problem there is that, yeah, one of the core principles of Bioconductor is interoperability, and then it's not very interoperable because it's not the default data structure for network analysis. What is the default data structure then? iGraph. So there's a package called iGraph. In the package iGraph, there's the iGraph class. So the iGraph class from the iGraph package. And this is what everyone uses for network analysis. So you have there the graphs themselves. So these are usually sparse matrices internally. You have node attributes and you have edge attributes there in the same object. So then, okay, so nice. We have the data structure iGraph. We have single step experiment. Let's combine them. So yeah, in single step experiment, you have call data, row data, metadata, and reduced dimensions. But then now we have 2 new slots, and these are row graph and call graph. Very similar to row data and call data in terms of design. So this is what you know, I guess, for those of you who know single step experiments. Again, assays, row data, call data, and reduced dimensions. But then now we have another slot, and that is row graphs, where you can store iGraph objects corresponding to rows of your assay, and call graphs, where you can store iGraph objects corresponding to columns of your assays. Okay, so this is what it looks like. So this is the constructor function. It's very similar to what you would do for single step experiments, but then you can have extra arguments here, row graphs. So this would be a list of iGraph objects, and call graphs, that would be a list of iGraph objects as well for the columns. And of course, if you know summarize experiment and single step experiments, they have to match. You cannot have things that are missing here and also in the assays. And this is the show method. So this is what it looks like. Everything of this is familiar to you, I guess. The last two rows are new. So the row graphs here. So this is one of the name of your row graph, and this is the name of your call graph. And you can have many if you want. You can also coerce, of course, from a summarized experiment object or a range summarized experiment or single cell experiment object. And then, of course, the row graphs and call graphs will be empty. If you want to access the graphs, this is similar to assay singular versus assays plural. So if you do row graphs in plural, you get a list of iGraph objects like this, row graphs and call graphs. And if you do row graph or call graph in singular, then you have the iGraph object itself. Again, similar to assays versus assay in summarized experiment nomenclature. The nice thing here is that row graphs are linked to row data, and call graphs are also linked to call data, which means if you have, for example, a graph here, so you do call graph once, you're getting the first call graph. This variable here, cell type, is a variable that was in call data, but was not in the original graph. But then these things are automatically propagated. So all the call data variables, they automatically go to node attributes of the graph. And same thing happens on the other direction. So for example, if you do row data and you have your graph experiment object, in this graph experiment object, there is a row graph called gene_core, so gene correlations. And then this graph had a degree vertex attribute. So this vertex attribute automatically goes to the row data. So you don't have to worry about getting all this information manually. Subsetting works as expected. So if you subset the entire object, you subset everything. So the rows, the columns, the row graphs, the row data, and everything else. This is just a demonstration. So yeah, we get the first 10 genes and the first 10 cells. And then you get an iGraph object with only 10 nodes. All right, so this is a package that I developed many years ago when I was still a master's student, and I regret some things that I did there, of course. I think that's normal that we do. That means that we evolved, that we learned. So one of the things that I did there is it's a package that does network inference. So one of the functions, for example, infers co-expression networks. And then, so these are a bunch of different edges that you have. So for example, you have correlations between genes, adjacency matrix. So this is like transform correlations, and then topological overlap as well. And then at the time, I used lists. So the output of the function was list of things, because I didn't want to have an overload of extra classes. But now I have a data structure that I think I can use to store everything concisely. So there is going to be, for those of you who use BioNeuro, there's going to be an update in the package coming to BioC Devel soon, where all of these things here will actually go to the corresponding, the right things, the right slots in the graph experiment class. So it's coming to Devel, and I've been in touch with other people, other labs, mostly in Belgium, where I'm based at, other labs in Belgium that have packages for network analysis, so GNU3 from Stijn Aarts lab and Nishinat from Ivan Sai's lab, so that they can also use start using graph experiment as a data structure, and then over time that this can become the default data structure. So some examples on how we can use this. So if you have transcriptomic data, for example, genes in rows and samples or cells in columns, you can use this to store co-expression networks, regulatory interactions, protein-protein interaction networks, and gene family information if you have any. And for the columns, this can be, for example, cell-cell distances and sample-sample distances. If you have proteomic data, it's very similar, but you would have instead co-abundance networks, we call, instead of co-expression. And yeah, everything else is the same. Now, if you have microbiome data, one of the things that I like here, so we also have co-occurrence networks, very popular in microbiome. But what I like here is genealogies. I guess you all have heard talks before on tree summarized experiment as a data structure for microbiome. And this is very nice because you can have the tree and the data together. But then the problem is, if you're working with different species, you cannot just, for example, regress body mass on longevity, because yeah, the data here are not independent. That's one of the assumptions of a linear model. You have to correct for phylogenetic distances. So species that are similar would have more similar values, and species that are more distant will have more distant values. Methods to do this, to correct for phylogenetic distances, are called PCMs, for phylogenetic comparative methods. And this is usually done with trees. The problem is, trees are very nice. I love trees. I'm an evolutionary biologist myself. But then, so yeah, they correct for these distances between so the species in columns here. But the problem is, trees don't work always, especially if you have what we call incongruence. So some sources of incongruence that we often see are horizontal gene transfer, so bacteria, check. Introgression, so if you have gene flow between different species, and if you have recombinations. So trees are always bifurcating. And then I don't think anyone should be using trees for bacteria. So it's a hot take. I'm sorry. But yeah, I don't even think there is such a thing as a bacterial species. And many people in microbiome, microbial would agree with me. So maybe we should be using networks here instead of trees. And then you can use graph experiment as the data class. With that, I would like to thank you all for your attention, and I'm happy to take questions.
Maybe we'll allow one question because we ran out of time. Any questions?
Thanks for the really nice and interesting talk. I know now I understand your opinion on trees, right? But is it still interoperable maybe with the tree summarized experiment class? So could you have both tree and graph?
You can have a tree and a graph because all trees, well, all trees are graphs, but not all graphs are trees. So a tree is essentially a directed acyclic graph with bifurcating edges. So yeah, but then you have to, of course, convert somehow from philo to igraph, but then you should have a function that goes back from igraph to philo. But yeah, essentially a tree is a graph, but you cannot have the philo object there.
Okay, yes. So like then tree summarized experiment should have like a way to work with digraph objects instead of
coerce, yeah. I didn't have coerce functions from tree summarized experiment because of the dependency hell that would add. Also, one of the reasons why I didn't, for example, extend spatial experiments. I wanted to have a spatial experiment extension, but then spatial experiment is very heavy in terms of dependencies.
Thanks.
Okay, so thank you again. Let's thank Fabrizio. Yeah, and now our next speaker is Giulio Benedetti from University of Turku, and he will present us Ariande, I think.
Hi. I think many of us got lost in biology while we were trying to do analysis. At least that's my case, but really nice introduction on graphs so far because we'll see them again. So let's say we have a set of microbes from a microbiome dataset. Can be something else, maybe genes, but let's stick to microbes. And we also have a set of food. And maybe these microbes, we want to know what food these microbes like. So one really easy way is to assume that all microbes like all food, but that's probably wrong. We want some way to get some meaningful knowledge about the links between the microbes and what they like. And one possible way is to use a reference database to understand what tools these microbes have. And with these tools, how are these tools then connected to the food itself? Maybe there is another reference database that informs us on, okay, with the fork, you can eat a burger and so on. But where do we find this information? Okay, they are in public databases, but can we access them? How? So there are very many databases on different omic types, UniProt with the proteins and similar protein sequence clusters. We have done KEGG, pretty famous, if not the first actually, doing these kind of things, integrating knowledge about the genomes and the organisms and the metabolic pathways performed by several organisms. Oops. And finally, reactome databases that contain information about reactions and enzymes. But these different databases don't necessarily have to be thought of separately from each other. They actually contain many related information across them. So turns out you can represent them as, you take the internal structure of these databases and represent it as graphs, where certain entities, biological entities, such as enzymes, proteins, genes, microbes, defined with their IDs, they can be present in different databases. So why not integrating this knowledge into a big databases? This way, then we can actually get something bigger than the whole, or the sum of the parts is actually bigger than the whole in this case, because we can then go beyond links defined by a single database. We can actually explore multi-database connections and go from something like microbes to, okay, what compounds they produce through what reaction and with what enzyme. But this is a bit complicated to do by hands, especially when you have 7,000, about 7,000 biological databases out there, and they all, most of them store information in different data formats. And maybe they have different ways to fetch the data. So the thing doesn't really scale up really well. That's why we asked help from Ariadne. She came for help. This is a new package that we plan to submit to Bioconductor. And what basically does is to provide a general framework to work with the database knowledge and find the pathway in this maze that is biology without you worrying too much about wrangling data to do it. So back to the graph that we made out of those three different databases, KEGG, UniProt, and RHEA. We can now go back to our original question. What each microbes, what food each microbe likes to eat, or maybe produce. Well, that's a different type of aspect here, but both are possible to search here. So we start with the NCBI tax IDs, and we can navigate to this graph and find the pathway that takes us from microbes to what GBI IDs that basically represent the metabolites, what microbes are linked to what metabolites using public knowledge from several different databases. And what you get out is something we call with Thomas a link map. That is basically a two-column data frame with the origin IDs on the left and the target IDs on the right, which gives you the links, the many-to-many links possible between those two databases, going through different resources. What kind of applications, how is this useful? A few examples are functional annotation of microbiome data. For example, using Chocoplan or Web of Life resources that link, for example, UniRef to KEGG orthologs. Another example is following functional annotation. You can then actually stratify your features, your microbial features of genes, into functionally meaningful groups, consortia of microbes that perform a certain operation, biological operation. You can do gene or microbe set enrichment analysis after annotating your data set with this kind of knowledge. And for that, there is M6DB, BUG6DB, and similar 40 ontology database, which is all integrated, by the way, in this package. And then pathway enrichment analysis using tools MetaSeq or the CAG metabolic pathways, and more. So if you can think about it, how to link a certain thing to some other entity in this graph, then there is probably a connection out there because there are so many databases. So to conclude, Ariadne is an attempt to provide a way to integrate public knowledge into this general graph and do it in an easy way, so that if you use this package, you don't really have to worry about where the data comes from or how to wrangle it so that it's compatible with your data set. Well, of course, it's important to be aware where the links come from, what the database links actually mean. But this can maybe be suggested in a way using this package. Two possible interesting applications are this feature stratification by some kind of biological knowledge and metabolic functional modules of microbes, and then pathway or set enrichment analysis. Your database that you're currently using for research is missing. There is actually a custom, a function to add custom resources to the graph. And if it's really something useful and conventionally used in your field, it can be added to the main package. You can just leave an issue. And that was it. So I really thank my co-developer and friend Thomas. We've been exchanging many ideas about this package, how to train it, and also other members from my group.
Thank you, Juho, for the talk. Are there any questions?
Yes. How do you deal with redundancy in databases? So if you look at LimROTS and CAG, I think they all have a single name for some information. So this is kind of redundant. How do you like that?
Yeah, there isn't a single pathway that takes you to microbes, to metabolic pathways. Those will be two different pathways, and you can choose which one to take.
So both of these, like the integral, for example, annotation will be from both databases independently, the graph.
Yes, those will be represented as two different edges. And you can, there are a few functions I didn't go into to explore this graph and see which pathway you want to take in the end.
Sorry, we are running out of time, so thank you again, Juho. And now our next speaker is Billy Oistintin from University of Turku, and he will present us the package not done.
So here to this R package that we use to process LCMS metabolics data. Okay, so maybe some of you already know about metabolomics. Actually, there should be an image here showing what it is, but it's somehow missing. So I'll just explain to you. Metabolomics is part of this omics cascade, and it sits closest to the phenotype, basically answering a research question, which is about what's happening in the metabolism right now in the organism. And liquid chromatography coupled with mass spectrometry is one of the key analytical methodologies to analyze samples in metabolomics, partially thanks to several benefits like high sensitivity and also dynamic range. But it also suffers from some complexities, let's say. So for example, in long sequences, the signal intensity can start drifting. There can be missing values for different reasons. A single metabolite can produce even more than a dozen different ions. And also some of these ions are not representing a true metabolite. So there can be some impurities. Also, we have to repeat the univariate statistics up to thousands of times for each data set. It creates some false positive bias in the data. And also we have this usual challenge in omics, how to visualize this kind of complex data and also the quality of the data. So Notame it's quite fresh in Bioconductor. It has been there available for about 6 months. And during the review process, we decided to divide it into a bundle of 3 packages based on their key functionalities. So this Notame per se is more for data quality assessment and correction of signal intensity drift, whereas Notame Stats is for running statistical models. And then Notame Viz is then for creating visualizations on the data. And these Notame packages, they sit in this overall metabolomics workflow somewhere between this aligned peak abundance matrix, which is in our pipeline generated by MS-style software, and the interpretation of the results. So it's trying to be basically do everything after the peak alignment, what is required, so that you get enough from your data to actually start compiling your manuscript. And it starts with the quality and then going to correction, clustering of molecular features, and then it goes to the statistics. And there's 2 different places where we mainly generate visualizations. Regarding the input that Notame wants, some of that actually should be taken into account already when you design your metabolomics study. So your LCMS sequence should be designed so that it allows quality management. It's very important to include extraction blank samples to determine what are the impurities in the samples, and also pooled QC samples that are kind of representing the overall sample, and these will be then injected every 12 samples or less. The actual input file that is put into Notame it can be basically an Excel file that has this matrix of peak areas across samples plus sample metadata and feature metadata. And this is an output that, for example, comes out of MS-style. So that's why we have kind of used this as the default output. If you use any other software, you might need to do some small changes in the input file. The first thing, of course, in LCMS metabolomics is the fact that if you put rubbish in, you get rubbish out. So it's important to assess the quality of the data. And we do that in this pipeline by calculating certain metrics of quality, for example, detection rate in the QCs, detection rate in the samples, relative standard deviation, dispersion ratio, and their robust versions, sample to blank ratio, and we have some default criteria for those. If any of these criteria are not fulfilled, there is going to be a flag given in the resulting data matrix. This doesn't mean that these should be removed. It just means that it's like a warning signal that something might be off with your signal, and typically we still manually verify whether this is true. The next feature that I will show you is the signal drift correction. So if you have a long LCMS sequence, what happens is that the samples start basically interacting with the instrument, and therefore we need to correct for this shift, which is kind of a little bit unpredictable, and it can change from feature to feature. And we use this algorithm that contains this cubic spline regression, and we also take advantage of these pooled QC samples that we have injected let's say every 12 or less samples. So we fit the regression line along the QC samples, and then we straighten the line, and then whatever is the difference, we then use to correct all the peak areas of that particular feature in all the samples, and we repeat this for every single molecular feature. Then another feature is this clustering of molecular feature. Like I mentioned, that a single metabolite can produce several different ions, which is a kind of like an annoying thing in mass spectrometry. It does that by combining this. So there's basically like two criteria. There's the coevolution and high correlation of features that is kind of signaling that they are coming from the same metabolite. So this feature is then clustering these molecular features based on this coevolution and then high correlation. It does a correlation network, and also it does a clustering based on the high mutual correlations within each cluster. And then finally you get this, let's say, tentative clusters that are supposedly then representing a single metabolite. So this will be helpful for the manual curation of the data. And just briefly about the statistics, so there's a separate package for doing those, which are typically used in metabolomics. So a lot of different univariate tests, multivariate tests, and correlation tests. And by default, we do the FDR correction for the good quality features. As you may know, it's also pretty much standard in for example, genomics and transcriptomics. So also in metabolomics, it's pretty much used. And then the visualizations package is doing, so basically it has like two main functions: do the visualization for the quality before and after the quality assessment and corrections, and then to visualize the results starting from overall image of the data like PCA into feature-wise plots like box plots. What we would like to do in the future, because this is still quite fresh in Bioconductor, we would like to add a feature that is making it easier for the user to select those features that then go to manual annotation, some kind of traffic light system based on multiple conditions. And of course, this multi-omics integration is getting increasingly important. So we would also like to benefit from this ecosystem to make some more multi-omics integration also using our package. To conclude, so Notame was still like developed for our internal use, so that those people who are not so familiar with or can use it easily, so it doesn't reinvent the wheel, it just makes it easier to drive. Notame has been incorporated as a functionality in MS-DAL5. And if you want to have some further look, give some feedback or just some contribution ideas, you can visit our lab website. There's also a book chapter preprint available about this package. And there was supposed to be an acknowledgements slide, but it has disappeared. But I anyways thank all the people who have contributed to this package, and thank you for your interest.
Thank you, Vili, for the talk. Are there any questions?
Thanks for the presentation. It's a very simple question. Basically, you had a lot of these parametric and non-parametric tests and models and whatnot. So does the package does these automatically, or the user basically it's a automatic gear or manual, you know, your car analogy?
I would say that you have to put like a manual gear because you have to select based on your study question that what are the appropriate tests that are needed, and also basically you need to make sure that your metadata is containing that information that you can then do the test. So there are some things that we do, like for example, some of this multivariate analysis you can do every time. So maybe that's something that can be done automatically, but basically study by study we separately decide which tests we do. Did you say normality? I'm not sure if it's like really included in this bundle. I'm pretty sure that it's like it can be like borrowed from other packages inside Bioconductor, but I'm not sure if we have it actually in Notame.
Okay, so actually there's no time left. Thank you again. Let's thank Lily. And now we will move on to the poster pitches.